Wow. All right. Thanks. So thanks for that introduction. So as was mentioned, my name is Gabriel LaHue. I'm a soil scientist down in Mount Vernon. And we're going to be talking, Deirdre and I are going to be talking today about a long-term experiment from the Soil Health Initiative that was started in 2021. And hopefully we're just going to provide some brief updates, try and increase awareness of this experiment, and hopefully this also sets the stage for a few talks you'll hear later on from some of our collaborators. So in addition to us, we're also working with Chris Benedict, who is putting on this whole event, Chakradar Matapalli, who will be talking shortly, and Louis Nottingham. And so I don't know how many, let's see. Sorry, give me a second to find how to advance slides.
Try using the keys.
Yeah, that doesn't work.
Okay, cool. Well, I am going to start with some announcements while they're figuring out those technical issues. So at the end of the session, we'll be asking you for feedback by text message. We'll have it up on the right-hand screen that says SoilCon Northwest Washington. We're going to provide, yeah, hey, I'm in the back. So you don't need to look at me, so okay. It's fine. So we're going to have those where you can text questions and also feedback. So we're looking for getting your opinions. It really helps us to improve the content of each year, which helps researchers successfully fund the projects that they're presenting on. So we really appreciate your time on that. Also is pesticide credits. This whole session is available for pesticide credits. And those are going to be in the back, but you have to stay for the whole session. If you're wearing a black hat and a red plaid shirt, you don't get pesticide credits. Just kidding. So that is it. And if you want to have any questions, I'll be here in the back. And then Deirdre and Gabe, go for it.
Great. Thank you. Well, I figured out how to advance the slides now, so we're starting off. How many folks have heard of the Washington Soil Health Initiative? Raise your hand if you've heard of it. Okay, that's actually a pretty good number. So for those that haven't heard of it, it's basically a tri-agency collaboration that was funded in 2019 in part and then fully funded in 2021. But it has the Washington State Conservation Commissions working on kind of the technical assistance piece, WSDA working on policy aspects and also kind of a regional soil health assessment, and then WSU really leading research efforts for the Washington Soil Health Initiative. And what we're going to zero in on is that figure you see on your right, which is this network of long-term research sites that was established as part of this soil health initiative. So there's 6 for the purposes of today. There's 2 that deal with potatoes, 1 in Othello that's just kind of getting off the ground, and then 1 in Mount Vernon that's focused on the northwestern Washington annual cropping systems. But really the driving crop in that rotation is the potato crop. And so Deirdre and I, when we first started here, we were tasked with really trying to flesh out this long-term experiment. And so one of the first things that we did is we had a soil health listening session. Some of you may have been at that session, but really not just potatoes, but kind of across the annual cropping systems, trying to get an understanding of what does soil health mean to people? What do they want out of soil health? What are the soil health challenges they're dealing with? And where are we now and where do we want to go? And that led to the Soil Health Initiative Roadmap. There's a chapter in there on the Northwestern Washington annual cropping system. So if you get a chance, I highly encourage you to check that out. But really, as 2 relatively new faculty members with WSU, tasked with starting this long-term experiment, it was invaluable to hear from folks. And so that their input really drove this, and they continue to drive that through our stakeholder advisory committee, which has, I think, about 8 to 9 members on it now. But if you're interested, feel free to reach out to us and engage with us, and we're happy to take additional feedback. But really, the 2 big challenges that we heard coming out of this were a degradation of soil structure, or tilth, just because of the intensive tillage required for potatoes, and then also soil compaction challenges, and then soil-borne diseases. So those are kind of the issues that rose to the forefront of the soil health challenges for the potato cropping systems. And with the raspberry session, we talked a lot about soil-borne diseases this morning, and you'll hear a little bit more about soil-borne diseases shortly. But you can just see, this is just a picture of after harvest this year, and you all know this if you're growing potatoes, but with our shoulder seasons and these soils, we really have a lot of challenges with reduced water holding capacity, slower infiltration of ponding, restricted root growth, surface crusting, all these things that can be exacerbated by these challenges of soil tilth. So using these, we set up a long-term experiment that we hope will go many years. It's currently just finished up its fourth year. And what that looks like is it's a gradient of disturbance and organic matter inputs. And so the idea was that we heard from a lot of other long-term experiments that it's really important that growers, we're never going to represent everything a grower does because everybody's system is different and they're never going to find their exact practices on here. But if we at least have a gradient, people might be able to say, find themselves on this system. So we have everything from kind of like our baseline worst case scenario for soil health, which is like full tillage. This is a 4-year annual crop rotation. So we have potatoes, and then we have a spring barley, a silage corn, and a winter wheat, which is a fairly common rotation down in the Skagit County. Obviously, everybody's rotation is different. There's some crops that we'd like to include in here that we couldn't, but really potatoes is the base, and we have all those crops represented in every year. But in our worst case scenario system, kind of for soil health, which is our baseline, it gets full tillage for all those rotations. It gets all the crop residue for like the grain crops baled off, and it doesn't get any compost or cover crops. And then we slowly start adding in practices like an annual cover crop and compost, reduced tillage. And then finally in kind of this most aspirational system 4, that's where we get actually between potatoes, it doesn't go into those rotational crops. There's no winter wheat or barley or silage corn. It just goes into a grass clover perennial cover crop for 3 years and then goes back into potatoes. And so that's really a system that's pretty different, but it's actually one that we're already seeing some folks are doing in Skagit County. We often get asked by folks like, could this ever be feasible? But it's like you start to see it out there on the land. So this is what that looks like. This is the aerial overview. As I mentioned, we have every crop in every year. So these long strips are, this is a potato crop, for example, and then in there it's subdivided into 4 so that you have like those individual treatments being imposed. And we're looking at a whole host of things. So we're looking at crop yield and quality, soil carbon stocks, a whole variety of soil health indicators on the physical, chemical, and biological sides, soil-borne diseases and pathogens, beneficial and pest insects, weed populations, and economic performance. You'll hear from Chakradar later about the soil-borne pathogens, and you'll hear from Louis later about, I believe actually Adriana, about the beneficial and pest insects. But today, Deirdre and I are just going to be talking about crop yield and quality and some initial soil health indicators. So just a quick spoiler alert that we haven't seen, we're still working on the data from this year, but we haven't seen any effects on crop yield from the first 3 years, which isn't entirely surprising. One, we want to make sure these treatments don't negatively impact yields, but also the whole reason we're doing long-term research in the first place is soils are slow to change. So you see a lot of studies that show, okay, we didn't see that much after 2-3 years of doing this. But really, it's about what happens after 5 years, after 10 years, because the soils are slow to change and we need to be looking at these management practices for the long term. So that's our yield on the y-axis and then just the year on the x-axis. You can see a giant dip in potato yield. That was 1 year where we had terrible surface crusting and it was just kind of spotty over the valley, depending on when fields were planted. Most growers would have tilled that field in, and a lot of growers did. But I think that will be something to return to that will be really interesting, because in those kind of adverse conditions, how does the soil health management practices impact that? So no effects on yield yet, but we are starting to see some effects on soil organic matter. And so that's actually pretty surprising to us already. This at the point this data was taken, it was only after 3 years of the trial. And just to orient you to the graph, because we have every crop in every year, this is just the phase of that crop that this was in that particular year. and here's soil organic matter, and then those 4 management systems. So this black dot, or the dark red, that's our system 4. That's our most aspirational system. And then it kind of goes down the gradient from there, where this green is kind of our worst case scenario that gets no crop residues, all the residues bailed off, no cover crops, anything like that. But we can clearly see that we're starting to increase organic matter just in the surface, the 0 to 30 centimeter depth. And you can see that we're getting that in these ones that have had the grass clover for longer. So because the rotation goes from potato to spring barley to silage corn to winter wheat, those that have had that perennial cover crop for longer, like the silage corn and the winter wheat, those are the ones that are starting to build up this soil organic matter. And it's really interesting to think about, the ones where we see it. This isn't just an effect of compost necessarily, or just an effect of the grass clover, because you do see in some cases higher soil organic matter in this one that has both. And we will be doing more complete measurements to get at the whole carbon stocks for the profile. And so with that, I'm going to turn it over to Deirdre to talk a little bit about some of the biological measurements.
Yeah, so as Gabe mentioned, we're doing this whole suite of chemical, biological, and physical measurements. And one thing we wanted to look at that is sometimes is quicker to change is microbial activity. And so one thing we measured was enzymes, which are produced by microbes and plants to break down cellulose, break down crop residues, and turn those nutrients into smaller compounds that microbes can take up, or plants can take up. So again, we're similar graph here where we show the different phases of the rotation. And you can see that in our treatment 4, that one that has the perennial grass clover cover crop in that treatment 4, so it's not actually in winter wheat or silage corn, it's actually in a grass clover. We are seeing higher levels of this enzyme beta-glucosidase, which is used by microbes to break down crop residues, basically. We took these samples in post-harvest, so one reason we may be seeing such a high level in this system 4 is that it was the only one that had living roots at the time. So as we move forward, it'll be interesting to look at different time points throughout the season, and then also relate these to other functions that we look for from the soil. So you can see that in the potato year, we don't see any treatment differences, but as we get further into this perennial cover crop, we are seeing higher levels of microbial activity. One thing that we're really excited, we just got data on Monday for data for this fall. And so it'll be really interesting to see, these plots went into potatoes this last season. So now we have post potatoes. And so it'll be really interesting to see if we see the continued higher microbial activity after that perennial cover crop has been turned in and that and a potato crop has grown. Similarly, this is looking at microbial biomass, similar time point, and we're seeing similar trends where we have higher microbial biomass. This is active living microbial biomass in the soil, higher microbial biomass in that system 4. Again, it could be due to the fact that there's living roots right at that point, even though these had just recently had living roots and had a lot of carbon inputs. So we're going to be taking samples again this spring, and it will be really valuable to look at the temporal effects too when all of the plots are kind of like ready for planting. So another thing that we're looking at is whether there's effects on nitrogen availability from the soil. Right now we've been fertilizing all of the plots the same and irrigating all of the plots in a particular crop the same. But one thing we want to do over time is think about, can we reduce inputs in some of the treatments that are maybe getting compost or getting more cover crops? Are they providing more water holding capacity or more nutrient availability? So this is showing data. This is a lab incubation. So soils were collected from the field, put into a lab incubation, and incubated at temperatures that were representative of the growing season. And so we did this twice, once in 2023 and once again in 2024. These were collected going into potatoes, so from those plots that had been in wheat or perennial cover crop the year before and were going into potatoes. And you can see that in 2023, we do see that treatment 4, we are seeing significantly higher nitrogen availability plant available nitrogen earlier in the season. So this could be a big benefit for the potato crop. We actually didn't see that there was greater cumulative nitrogen availability, but more just more earlier in the season. We're not seeing the same trend this year in our lab incubation, but we're continuing to do it. So you can see we're not quite done with the lab incubation for this year. So it'll be interesting to see whether we see the same trend for the repeated lab incubation. Yeah, and this is just saying that the rate of nitrogen release is likely faster. I guess I had already said that. So going forward, there's a lot of questions we can continue to look at, and we're really interested in whether the disturbance from the potato phase kind of brings things back to the same baseline as all the other treatments, or whether the benefits that we're seeing from some of the compost and the cover crop are continuing through the potato season. And just to know, this was an earthworm that we actually saw during potato harvest in a treatment for potato crop. So we anecdotally saw more earthworms in the potato fields that had been in that perennial cover crop the year before, which is pretty exciting. We're also thinking about this perennial cover crop and, are there alternatives that we could use that might yield some cash value? So could a perennial wheat crop be used, or potentially alfalfa be used instead of the grass clover? And you'd still get that benefit potentially from the perennial cover and the carbon inputs, but you would also get some yield from it as well. So as we mentioned, we're just finishing year 4, and every 4 years we do a big extensive sampling, which we're going to be doing this coming spring. And so we'll have, we're going to be going down to 4 feet in depth for our soil sampling. So we'll really be able to even characterize even more the changes in the soil that we're seeing. And then we're also going to be looking at, economic analysis. And we're hoping to expand this to on-farm trials where we can look at some of the treatments that we're doing in our controlled trial, but look at it in other soils and other contexts in the area as well. And with that, we're happy to take any questions. And so as Gabe mentioned, we're going to be hearing from the potato pathology group next, who's been working on this trial as well, and then also from the entomology group. So we're collaborating with this and trying to look at these systems from a variety of aspects.
Thank you very much, Gabe and Deirdre. Question for you, Deirdre, regarding that nitrate test that you did there. Can you explain a little bit more why you didn't see an increase in the cumulative nitrogen that came out of T4, and whether there's maybe nonlinearity in how that's measured in the assay? Or I imagine there's more to it than what you presented.
Yeah, that's a good question. So I don't have any concerns about the detection limit, that it's getting too high or anything in terms of the amount of nitrate in the soil. It could be that there's, and I'll see what Gabe thinks as well. It could be that maybe in the incubation itself, there's a buildup of nitrate, which could be leading to like denitrification. And that's something we've tried to control for by not having the soil be too wet. And trying to kind of find that sweet spot. But that could be happening, that we're getting some loss of nitrate through denitrification. And I don't know if Gabe, you want to weigh in?
Yeah, I mean, we did look at various moisture contents to control for that. But at the same time, we all denitrification can happen in those really anaerobic microsites, and I think there's been a lot of work on that recently. So I think we're trying to minimize it, and we did kind of optimize the water content in order to be able to minimize it. But it's a good question.
Question over here.
I see that you're adding the compost to some of your treatments. I was curious if you've considered, I just had a, I just lost it. The charcoal, yes, if you consider it, because I know that with some of the studies I've seen, it can take several years before the impact of mixing that into the compost is seen in the resultant plants. So I was curious if you consider doing that.
Yeah, we hadn't considered doing biochar. With the compost, we are just trying to kind of represent an organic matter amendment, and not many growers in our area in Skagit are using compost, but a lot of them might be using manure. And we thought about using manure, but not everyone has access to manure. So we're using a yard waste compost that we're hoping, could be something that might become more available. We didn't consider using biochar just because it's has a lot of other properties that could influence things in different ways, but we're really just trying to get more carbon into the soil with the compost amendment.
All right, with that. We're going to move on to our next speaker. Deirdre and Gabe will be around for more questions. So next up, we have Christina Alcania with Washington State University, and she is an agricultural research technician.
Hi, everyone. Yeah, my name is Christina Alcania Gallegos. I'm representing the Berry and Potato Pathology Lab with the Northwest Research and Extension Center down in Mount Vernon, Washington. Today, I'll be talking about the impacts of reduced disturbance and organic matter additions on potato soil-borne pathogens. So Gabe and Deirdre already explained to us what the ELTER project is about. In case you didn't catch it, ELTER stands for Long-Term Agroecological Research and Extension. And as I mentioned as well, our site in Mount Vernon looks at the potato-based annual cropping systems. The general objective of this project is to measure the impact of conservation practices on soil health and farm profitability. Another more specific aim is to look at disease incidence, and we're trying to answer the question, are pathogens present, and if they are in populations that affect crops quality. In this instance, the crop refers to potatoes. Once again, Gabe and Deirdre already explained to us what the treatments are about. But what I want to mention here is that many plant diseases affect potatoes, and to help minimize the loss to diseases, farmers usually use a 4-year crop rotation, and they use the 3 seasons in between to try and to reduce the amount of diseases infecting potatoes. So these 4 different treatments try to mimic what growers do in their fields. And as it was already explained in the previous presentation, all these 4 treatments have different levels of soil disturbance, crop residues, annual cover cropping, and soil amendments. Now, I'll briefly run you through our sample processing. For the ELTER project, we soil sampled the potato plots. We did this by randomly sampling soil from 10 spots at a depth of 0 to 15 centimeters. Then we bulked these samples into a bucket. We took these soils back into the station. We let them dry for a few days, and then we measure 250 milligrams of soil for DNA extraction purposes. This allows us to later quantify the amount of pathogen present in the sample. Then we moved on to qPCR, which stands for quantitative polymerase chain reaction. It's a very long name, but it basically allows us to detect if a pathogen of interest is present in the sample. And with those numbers, we run data analysis to determine if these effects are significant. So at our lab, we work with a few soil-borne potato diseases. One of them is black dot. This is caused by Coleotrichum coccodus. This is a common disease that affects the skin quality and marketability of potatoes. It can be found on stolons, roots, and tubers and leads to plants being wilted. Once black dot enters a field, it can survive for up to 7 years, and it also can continue to grow in storage with warmer temperatures. We also look at silver scurf caused by Helminthosporium solani. Silver scurf produces blemishes on the tuber surface, which lower the value of the crop in the fresh market. Lastly, we also work with verticillium wilt caused by Verticillium dahliae. This pathogen causes the following symptoms. It causes unilateral stem wilting, as well as browning of the stems and tubers. These damages lead to early crop senescence, as well as reduction in tuber size and hence yield losses. Once verticillium enters a field, it can stay there for up to 10 years, so it's kind of hard to manage. There are no potato varieties that are resistant to it, but this can be prevented with careful crop rotations. Some of the most common mechanisms for managing soil diseases in potatoes are disease prevention, suppression, and control. Prevention refers to the flora of the land. And so arid or virgin lands are more susceptible to diseases because there's no microbial competition. If a disease enters a field, then we move on to suppression and control. So with suppression, this can be naturally occurring or influenced by management practices. It refers to microbe-based protection that the plants have, and therefore the pathogen does not persist. Lastly, we have disease control, which can be achieved through the addition of organic amendments and crop rotations. So the Elter treatments incorporate all of these factors at different levels, and I'm not going to talk much about soil disturbance because we know about the benefits of minimal soil disturbance, but we also know that's not an option for potato. So, but what about the expected effects of the other elements? So for crop residues, these provide a continuous source of feed for soil microorganisms and increase abundance and diversity of microbial populations. For cover cropping, there's the potential for producing compounds that suppress pests and that can act as biofumigants. Both of these are disease suppressive capabilities. As for soil amendments, it can help to inoculate the soil with microbial diversity and suppress pathogenic populations. This is a type of disease prevention strategy. So what does the literature tell us about the effect of increasing soil organic matter on these pathogens? And in here, I'm only going to talk about verticillium because, as you'll see, this is the only pathogen that we were able to detect in our soil samples. But the literature tells us that verticillium readily colonizes sterilized soil, but has very limited activity in natural soil. Also, that adding animal manure contains volatile fatty acids that are responsible for the suppression of certain diseases, including our pathogen of interest, which is Verticillium dahliae. Fish emulsions also have a similar effect and can also help to combat common scab of potato. Another one of the elements of the elder treatments is the crop residues. And so one study showed that incorporating millet, for example, into soil had little effect on the reduction of verticillium wilt, though it helped with yields. However, incorporating broccoli helped to reduce verticillium dahliae populations, and this is thanks to the release of compounds called glucosinolates. So with all this knowledge, all this background, we know that soil-borne diseases are best managed with increased soil microbial diversity, which is accomplished by cover cropping and organic soil amendments. Therefore, our hypothesis for this study was that the lowest disease incidence would be expected under the Elter treatments that incorporate these factors, which are treatments 4, 3, and 2 in that order. So our preliminary results for soil data, for the past 3 years, we did not detect pathogens causing silver scurf or black dot. However, we did detect verticillium wilt. We want to know if the treatments had any effect on these numbers. However, when we look at the variation within each year and within each treatment, we cannot really say that this can be attributed to the treatments. Moving forward, though, we might see effects become significant. As it was already mentioned, next year it'll be the next full rotation. So by next year, it'll be the second time that potatoes go back to a plot. Besides soil sampling, we also quantified internal and external defects of potato tubers. We observed symptoms for black dot, black scurf, and common scab of potatoes. So in 2022, black scurf incidence was very low, ranging from 2 to 9%, whereas common scab ranged from 44 to 57%. In 2023, black dot incidence was 11 to 25%, and common scab was very low, 2 to 6%. Again, there were no differences. These differences cannot be attributed to the treatments as of now. So yeah, in conclusion, the LTER will keep looking at the legacy effects of each treatment on disease incidence for soils in the long run. And once again, acknowledging that it takes time to change the biological and chemical properties of soil. And you already know the funding agencies, and yeah, thank you very much. Please let me know if you have any questions.
We have time for maybe one question, one or two. Speaker is Louis Nottingham. He's an assistant professor in entomology with Washington State University.
Thank you all for being here. So today I'm going to be talking about the entomology side of the LTER. So just in case you just showed up, you need a little background on the LTER. This is the Long-Term Agricultural Research Extension Site in Mount Vernon. This is based around, or it's a potato-based crop rotation. So potatoes need to be rotated out of potatoes for various reasons. So with that, you have a lot of choices that you can make in what you want to do in those years when you're not in potatoes. And those choices can affect the soil quality. So how can we do that? Well, if you have different winter, or if you choose to have a winter cover crop or not have one, what kind of crops you're growing in the off season, if you're adding things to the soil, and then how much you're actually disturbing things, whether that be through tillage, through pesticide use, through using long-term cover crops or not. All those things can affect soil quality, and that is the main reason behind the LTER. I'm not a soil scientist, I'm an entomologist, so why am I talking? Well, these things can affect insects and bugs in general as well. And we want to understand like what kind of effects those might have. So we have a lot of important arthropods and other things that we have to study as entomologists, including slugs and things like that, that can be affected by all this. So we have pests, we have natural enemies, pollinators, decomposers, and even some things that, you know, they're just neutral to us, but maybe they serve as a prey source to things that are beneficial. So I'll just give a couple examples of why our choices in the LTER may be important in terms of insects and the different ways that we can kind of look at this. So crop choice is one way that we can start to look at what can go differently here. So by choosing certain crops when you're outside of potatoes, you can either increase or decrease bugs that may be good or bad, and they can affect you in your field at that time, maybe you in that field later, or even your neighbors. So who knows what this is? If you grow potatoes, you better know. Click beetle, right? So, and the click beetle is the adult of the wireworm, the notorious wireworm, tough pest in potatoes and a lot of other crops. So the interesting thing about click beetles and wireworms is that they come in and they lay their eggs normally not on potatoes, it's on some other crop, like tend to be grasses, but can be a lot of other things, even just pasture, that kind of stuff. And then the larvae stay in the soil for multiple years. So what you do in the off season can have an impact in the current season. And they can have a good or a bad impact. You may have a repellent crop as opposed to something that attracts it. So you can also attract natural enemies. So here's a picture of a couple of parasitoid wasps. Both of these are known to attack our most common potato aphid, the green peach aphid. And these things love nectar and floral resources. So if you have a field that is in a cover crop, a long-term cover crop that's flowering, and then you have a potato field next door, you may be creating a refugia or a nice little home for all these beneficial arthropods. And there's other beneficials. So everyone knows the friendly bumblebee. So if, let's say you, again, have an area where you're growing flowering crops. If you have neighbors that are also in, maybe in blueberries at the time, you could be supporting pollination for those folks, or maybe you also grow blueberries and this is helping you out. So those are crop choice impacts. There's some other ways that we can impact arthropods. So